Hey everybody, let's talk about the DNA replication mechanism. And I'm going to divide the mechanism into 6 steps, and as we go over this mechanism, we are going to talk about 2 enzymes, DNA polymerase 1, DNA pol 1, and DNA pol 3. And sometimes I'll just call this one pol 1 and this one pol 3. And there are some properties of these enzymes that are important with respect to DNA replication. So, initiation of synthesis. So neither one of these can initiate synthesis. So we're going to need to use, talk about an RNA polymerase to initiate synthesis. So both of them can synthesize into, in the 5' to 3' direction. So 5' to 3' synthesis. Yes, they can both do that. 5' to 3' exonuclease activity. Only DNA pol 1 has that activity, and I will describe what that activity is used for in a moment. And 3' to 5' exonuclease activity, and they both have that activity. And this is for a proofreading or error correction property of both enzymes. So keep these in mind, and let's talk about the mechanism. So 6 steps. First step is going to be DNA is unwound by a helicase. Okay, so what does that mean? Well, we're going to talk about replication in E. coli, and as we know, E. coli has a single circular chromosome, and that circular chromosome has an origin of replication. And replication is going to occur bidirectionally. So at the origin, from the origin, replication will occur both to the left and the right. So replication is bidirectional. Now, let's say this is the origin of replication. Doesn't look much like an origin, does it? Or DNA even, for that matter. So here is the DNA molecule, and I'm showing it as a double helix. So that's my diagram of a double helix, and it goes on. So let's imagine this is just a zoomed in version of the origin of replication. Now the helicase comes in and unwinds the DNA at the origin. So we end up with something like this. So we can call this a replication bubble. And I'm going to put a helicase right here. And I'll put one over here. Now the helicases will continue to unwind the DNA as replication progresses. So this side right here, this is going to be a replication fork. So this is like a fork in the road right here. This right here, we'll call this a replication fork. And one fork moves that way, and the other fork is on the other side, and it moves in the opposite direction. So that's the bidirectional nature of replication right there. 2 replication forks moving in opposite directions from the origin of replication. Now, for this replication bubble right here, we are going to have these small proteins called single-stranded binding proteins, or single-stranded DNA binding proteins, SSBP for single-stranded binding proteins. And we could call them single-stranded DNA binding proteins, SSBPs. Now these will bind to the single strands of DNA, and they will keep them from coming back together. So that's step one. DNA is unwound by the helicase, and then we have all this other stuff happening, right? Okay, the single-stranded DNA binding proteins bind and keep the strands from coming back together. We have the replication forks established. We have the helicases parked at the replication forks. And the helicases are able to keep unwinding DNA as the replication forks move away from the origin. So step two is essentially step two, I have it written down here somewhere. And so the tension is going to be reduced in these strands right here. So as the helicase unwinds the DNA, tension tends to build up down here. And I showed a YouTube video in lecture on this process. It's like if you have a wound rope and it's held still at one end, as you pull the rope apart, tension builds up and it becomes really hard to unwind that rope anymore. And the same thing is thought to happen with DNA. And so we have an enzyme called DNA gyrase, or just gyrase. We can call it DNA gyrase. And what it does, well, it's a special type of enzyme called a topoisomerase. And what these do is they'll cut one strand and they'll unwind the DNA a bit, and then they'll reseal the cut strand. So it will release the tension. So the replication fork can keep moving forward and the DNA can keep being unwound by the helicase. So there'll be a gyrase on both sides ahead of the replication fork, releasing the tension as it builds up. So that's step two. I'll just put DNA gyrase reduces tension. So step three has to do with that property of DNA, that first property we talked about with respect to DNA polymerases. Remember, they cannot initiate synthesis. So step three is RNA primase. Sometimes it's just called primase. RNA primase makes RNA primers. So let's go back to here for a second. Now, for the rest of the mechanism, what I'm going to do is I'm going to focus on just this fork, this replication fork over here. Just note that whatever we talk about over here, the same exact thing is happening over here on this replication fork. So this replication fork is moving to the right, this one's moving to the left. The same stuff that's going on over here at the right is also going on over at the left. So I'm going to zoom in on that replication fork. And I probably could have taken more up of the page up, so let me diagram this again. Step three, RNA primase makes RNA primers. So let's try that again. Okay, there's the replication fork. Here's the helicase. Here's the wound DNA. Here's gyrase, releasing the tension. Now, let's say this is the 5' end of a DNA strand, and this is the 3' end of this strand. So you should be able to determine whether to put a 5' or a 3' here and a 5' or 3' here for the end of this strand down here. So if there's a 3' down here, well, you know the 5' should be down here. It gets a little complicated, right? Because I drew it twisted like this. But imagine it wasn't twisted for the sake of knowing where to put the 5' and 3'. So if there's a 5' here up top, then there should be a 3' up top right there. Now, RNA primase also synthesizes DNA, or RNA, I should say, RNA in the 5' to 3' direction, but it does not need a primer. It can initiate synthesis on just a naked DNA template. So I'm debating whether or not I should show or diagram the nucleotides here. So, okay, so RNA primase can come in, and let's say it puts a primer right here. So that's the primer, and this is the 3' end of the DNA. This is the 5' end of the primer. And these are ribonucleotides, and this is an RNA primer. And let's say it's 8 to 12 nucleotides long. So this is an RNA-DNA hybrid. These are RNA nucleotides, these are DNA nucleotides. So if this is a U right here, then we know this is an A right here. This is a G, and this is a C. Now we have a 3' OH right here at the end of this primer. And in the previous video, we saw how DNA polymerase uses the 3' OH, so that 3' carbon of the last nucleotide has a hydroxyl group on it, uses that to connect it to the next nucleotide. So let's say this right here is also an A, and this is a G. Well, DNA polymerase can now come in here, because this RNA primer is here, it can now come in here, it can add a T, it can add a C, and so on and so forth. Whatever these bases are down here, now DNA polymerase can come in and it can extend this primer, polymerize DNA, based on the complementary base on this strand right here. Now, let me talk more about these strands. Well, let me put the primers down. We're going to get a couple primers here. So RNA polymerase can put a primer right here. So we have the 3' OH here, the 5' end right there. So let me put RNA primer there, and let me put another primer right here. RNA primer. So primase is busy, it's putting RNA primers once over here, and let's say multiple times on this one. And I'll explain why we have multiple primers on that strand. So at this replication fork, you can see we have 2 single-stranded DNA elements, and I could diagram the single-stranded binding proteins here, but to make it neat, I won't put the SSBPs here all along the strands. We're going to call this single-stranded DNA, we're going to call this the leading strand template. And we're going to call the DNA strand that's being made on this template, we're going to call this the leading strand. Now, the leading strand is formed by continuous synthesis. And we say it's formed continuously because the DNA polymerase here, now I should also say, DNA polymerase 3, this is the primary enzyme doing most of the DNA synthesis. DNA polymerase 1 is going to come in a moment. DNA polymerase 3 is doing most of the synthesis, and we say the leading strand is synthesized continuously because we have this one RNA primer here. DNA polymerase can find this 3' end with the 3' hydroxyl, and it can start adding the DNA nucleotides. And what it can do is it can follow the replication fork. So DNA polymerase 3 can follow right behind this helicase here, and it can just follow the replication fork. It never needs to stop synthesizing DNA because, you know, it's going in, the replication fork is moving in the same direction as the DNA polymerase. And if that's a little confusing, you might be able to understand it a little better when we consider this other strand up here. So this other DNA strand, we're going to call this the lagging strand template. Now, in these lagging strands that are going to be made, well, we can call each of them lagging strands. Oh, the whole thing is going to be called the lagging strand, I should say. So the lagging strand is going to be this one. And it's called lagging because the DNA polymerase 3 has to move in the opposite direction to the movement of the replication fork, right? So synthesis has to happen from the 5' to 3' direction. So this is the 5' end of the lagging strand, you know, starting with an RNA primer for now, and it's moving towards the 3' end. So synthesis is moving in the opposite direction as the replication fork. So we're going to call this the lagging strand, and it's lagging because it needs to start, stop, and then start again. So as this moves, we're going to have an unsynthesized piece of DNA be available for RNA primase to come in and make a primer, and then DNA polymerase 3 to synthesize that little section that just opened up after the replication fork moves to the right. And it has to do that over and over and over again. And that's why this is called the lagging strand, and that's why this strand is called the lagging strand template. Additionally, these little fragments that result because of the lagging strand synthesis mechanism, making these little segments, they are called Okazaki fragments. So long story short, the lagging strand template is synthesized as a series of small fragments called Okazaki fragments. And it has to be synthesized that way because the polymerase only synthesizes in the 5' to 3' direction, and with respect to the lagging strand, you know, synthesis occurs in the opposite direction to that of the movement of the replication fork. So you can see now a problem. So that's step 3 is RNA primase makes RNA primers. Step 4 we already covered. Step 4 is the leading and lagging strands are polymerized or synthesized. Polymerized and synthesized, they mean the same thing with respect to replication. So now we can see a problem, right? Okay, this strand looks pretty good. We do have to remove this primer up here eventually, but we have a bigger problem with respect to the lagging strand, right? The lagging strand has lots of these RNA primers here, and plus these ends right here, this end, which is DNA, the last nucleotide made by DNA polymerase III, when it runs and encounters this nucleotide right here, this RNA primer, it can't do anything. Why? Because initially, remember how we talked about how DNA pol 3 lacks a 5' to 3' exonuclease activity. So DNA polymerase III lacks a 5' to 3' exonuclease activity. What does that mean? DNA pol 3 is synthesizing in the 5' to 3' direction. It bumps into a ribonucleotide here of the RNA primer of the downstream Okazaki fragment, and it can't do anything. It can't remove it. So it falls off the template and essentially leaves a nick in the DNA strand. Okay, now DNA polymerase I has to come in here, and it can use the 3' end of the last nucleotide, and it can remove the RNA primer because DNA polymerase I, it possesses the 5' to 3' exonuclease activity. So let me diagram that for you a little bit better. Step 5, and I think I have this, I want to make sure I have this. Yep, step 5. RNA primers are removed by DNA polymerase I, and gaps are filled. Okay, so let's say this is the lagging strand template. Now in the previous diagram I had it at an angle, but I'm just going to diagram it as a horizontal line right here. Say this was the 5' end of the template, this was the 3' end, and let's say this is an RNA primer right here, and let's say DNA polymerase III had already extended that primer, and this segment of the chromosome has already been replicated. I'll shade this in to indicate this is the RNA primer. And let's say there's another fragment up here, but this is just DNA. I won't show the RNA primer up there. And let's say this is the 3' end, right here. This is the 3' OH of the last nucleotide that was laid down by DNA polymerase III. Okay, so DNA polymerase I, it's going to enter here, use that 3' OH, it's going to put down DNA nucleotides, and it's going to remove that RNA primer. So DNA pol 1, it can chew out that primer and put down new nucleotides, DNA nucleotides. So after DNA polymerase I comes in, we should end up with something like this. And there's one mistake in this diagram. I'll point that out in a moment. Now, why does E. coli, not just give DNA polymerase III, or why did DNA polymerase III not evolve a 5' to 3' exonuclease activity? It seems like it would be much more efficient. I don't know the answer to that. I haven't heard or haven't tried to look up an interesting hypothesis to why that's the case. I'm going to try to do that in the future. But for now, we'll just know that E. coli does use 2 different enzymes for this process. It needs DNA polymerase I to remove those primers. And after DNA polymerase I removes that primer, fills in the gap by adding DNA nucleotides there, it kind of looks like this. Now there's one mistake here. So when DNA polymerase I finishes, and it may fall off right after the RNA primer has been chewed up, or maybe it extends a little bit more and chews off and removes some of the DNA nucleotides, and then puts down new nucleotides. when it is done, there are no RNA nucleotides left. But when it falls off the template, it does leave what we call a nick. So all the nucleotides are present, but there is a 5' phosphate on the downstream nucleotide. So a 5' phosphate means the phosphate is attached to the 5' carbon of the downstream nucleotide. So that the nucleotide immediately downstream of the position where DNA polymerase 1 fell off the template, and a 3' OH group from the last nucleotide it added to the template. So this is called a nick. When all the bases are present, but there's no phosphodiester linkage between the two bases, that's called a nick. And what has to come in here is the final step of replication. And that is DNA ligase has to seal all the nicks. DNA ligase seals all the nicks that are present in the lagging strand because of removal of that RNA primer and filling in of the gaps by DNA polymerase 1. So it leaves behind a bunch of nicks, and DNA ligase is an enzyme that comes in and seals all of those nicks. So DNA ligase will seal that nick, and then we have essentially completion of replication. Now again, completion of replication after we have our original picture, after the forks have completely come around and replicated that whole chromosome. Replication ends approximately halfway down here. The termination process is somewhat complicated, and we are mostly going to focus on just how replication occurs and not worry too much about how it terminates down here about halfway through the chromosome. Okay, those are all the steps of DNA replication in E. coli. We will touch briefly on the mechanism of replication in eukaryotes. It's very similar, just a few differences we'll need to address next week in lecture. Okay, so as always, if you have any questions, I hope you come to office hours. I hope you email me with the questions, and if I don't hear from you, I hope you enjoy your weekend, and I will see you next week in class.